So our case study was on cystic fibrosis. So cystic fibrosis is a genetic disease that causes extremely severe damage to the lungs, and that is caused by the mucus buildup from the mutation of the CFTR gene, which is going to be later discussed in this PowerPoint. And cystic fibrosis is one of the most common genetic disorders in the US. Over 30,000 people have this disease. So the first question is, what are the causes, treatment, control, and prevention of cystic fibrosis? So the causes and diagnosis. So it is caused by a genetic mutation, and each parent can carry one of the CFTR genes, so it can be passed down to the next generations. This disease can pertain to respiratory symptoms and digestive symptoms. In order to diagnose this disease, usually a test by sweat sample is run. So it's usually done on children, and the doctors will take a sample of the sweat and look at the levels of salt. And if there's a high level, it usually indicates cystic fibrosis. So for treatment and control, there isn't a cure found for cystic fibrosis, so it is just treated by medications, antibiotics, chest physical therapy, and exercise. Those are just the top three, but these just help regulate the symptoms, and chest physical therapy will help loosen up that mucus that's in the lungs. So prevention. So since the disease is caused by a gene mutation, it can't necessarily be prevented, but usually gene therapy at an early age could repair or replace a defective gene. In this case, it would be the CFTR gene.
All right, so question 2. How would PCR be used to investigate the case of cystic fibrosis in humans? Consider all of the steps in the PCR process. So I'm going to assume that the audience has no idea what PCR is and go over the basics. So PCR stands for polymerase chain reaction. The basic idea of PCR is to copy a small amount of DNA rapidly. Depending on a person's job, PCR can be used for loads of different things. PCR can be used to help diagnose various diseases by being able to locate a pathogen or come up with some kind of genetic test so people can see if they are positive for some kind of genetic disease. I actually did an experiment with some genetic testing in high school where I had to use PCR to analyze fly genetics. So that was a really cool experiment. And then sometimes also in forensics labs, scientists may not be able to test on such a small amount of DNA, and then they'll put it through a PCR chamber and go over, whose DNA it is and possibly find a culprit in some way. So how does PCR work? So I'm going to explain the basics of it, not entirely the crazy process that goes through, but just the bare bones of it. So before PCR can even begin, a microtube needs to have 5 ingredients, which include a DNA template, a primer, a DNA nucleotide base, a buffer, and an enzyme. The buffer and enzyme will be selected specifically to achieve the desired result, hence why I did not mention a specific one. Once all 5 are in a microtube, the microtube can go into the PCR machine. I have a little picture of it in the top right corner. The PCR machine can run for a long period of time or a short one. It just depends on the desired result. And the PCR machine is going to go through a rapid thermal cycle in order to multiply the amount of DNA in the microtube. The thermal cycle is going through 3 phases until the timer is up, and those 3 cycles are denaturing, annealing, and extending. Apologies if I pronounced those wrong. Anyway, denaturing is when DNA is heated up enough to separate into a single strand. Annealing is when a temperature is lowered to allow the DNA primers to attach to the template DNA, also now known as the single-strand DNA, as it's been separated through the denaturing stage. And then finally, extending is when the temperature is raised and the DNA strands reconnect because of the enzyme, hence why you need the 5 things listed before to have PCR work successfully. In the bottom right corner, you can see a little diagram explaining on some of the temperatures and Just a little bit of a visual representation of PCR. So, how can PCR help with cystic fibrosis? Well, for starters, PCR can detect if someone has cystic fibrosis. The gene they are specifically looking at is CFTR. If that gene is mutated, most likely that person has cystic fibrosis. However, PCR is able to detect if there's a mutation on the cystic fibrosis gene. Surprisingly, there are over 2,000 identified mutations of cystic fibrosis, which means not all cystic fibrosis is going to be the same for everybody. There's lots of variations. Using PCR to identify what form of cystic fibrosis someone has is honestly the best way to do it, as it's most likely cheaper, well, it's not most likely, it is cheaper than many other methods, and that's because PCR requires such a small amount of DNA, and it also, the amount of primers, buffers, enzymes needed is so little, it's much cheaper in comparison to a lot of other genetic testing methods. And because we have been able to identify 2,000 mutations of cystic fibrosis, PCR is very successful in being able to determine what form of cystic fibrosis someone might have.
Question 3 asks how to differentiate between a cystic fibrosis carrier and an affected person through alleles. Distinguishing characteristics can be found in both the genotype and signs and symptoms of people. People with only one copy of the cystic fibrosis genes are carriers, meaning they do not experience symptoms of the disease. A homozygous recessive genotype indicates cystic fibrosis, and a heterozygous genotype indicates the person is a carrier, meaning they carry the recessive allele but don't present symptoms. Further distinction can be made through signs and symptoms. Carriers do not experience symptoms as affected people do. However, symptoms include frequent lung infections, persistent shortness of breath, and nasal polyps. Genetic testing can be done in order to diagnose people with CF. This testing entails taking cheek cell or blood samples.
Hi, I'm Lila. I'm going to be presenting my part of the presentation. So basically, according to UCSF Health, an individual must inherit 2 non-functioning CF genes, one from each parent, in order to actually have CF. So this means that in order for a child to inherit CF, they have to receive both recessive alleles containing the CF mutation in order to actually get CF. So even if both parents do not have CF, it is still possible that they can pass it down to their kids, but both kids do need to have both recessive alleles. Alright, so when creating the Punnett square, capital C, is going to represent the dominant normal allele, and little c is going to represent the recessive allele with the CF mutation. So if both parents contain the recessive allele with CF but do not have CF themselves, they're still going to pass the gene down, giving their children that chance of CF, as I mentioned before. So by creating a Punnett square, we can figure out that their children will have a 25% chance of inheriting CF if you look at our little Punnett square over here. They would only inherit CF again if they inherited both recessive alleles. So the phenotype would be 1 homozygous dominant without CF, 2 heterozygous without CF, 1 homozygous recessive with CF. The genotype would be 1 capital C, 2 capital C lowercase c, and 1 lowercase c. And again, that 1 with the 2 lowercase c's is the one with the 2 recessive alleles, and that would be the only percent chance that they would have CF if their parents were heterozygous with that CF gene. So again, the probability of having CF for the children, the F2 children generation, would be 25%. All right, that concludes my section. Thank you.